Hello friends, welcome back. And today we are going to study the topic developmental disturbances affecting the structure of the teeth. Under this category, we are going to study about enamel hypoplasia or hypoplasia of enamel. This could either be induced genetically or environmentally. If it's induced genetically, then it's known as amelogenesis imperfecta, and if it's induced environmentally, then it's known as environmental enamel hypoplasia. Then we are going to study about dentinogenesis imperfecta, dentin dysplasia, and regional odontodysplasia. Okay, I'll be discussing in detail about all these topics in my coming up lecture. So today we are going to study about amelogenesis imperfecta. With the name, amelogenesis stands for enamel formation and imperfecta stands for imperfect. So amelogenesis imperfecta as a whole stands for imperfect enamel formation. Now let's look into the definition of amelogenesis imperfecta. It is a congenital genetic disorder. Now, when we say it's a congenital genetic disorder, one thing we should make very clear in our mind, and that's not all congenital disorders are genetic in origin. Congenital means a defect which was present at birth. So, the defect could either be caused by environmental factors like injury, even. But amelogenesis imperfecta is that disorder which is congenital genetic in origin and it causes structural defects in the enamel. Now, this sentence over here is very important. As soon as you hear the word amelogenesis imperfecta, this sentence should strike your mind, and that's structural defects in the enamel. Okay, so amelogenesis imperfecta is that disorder which causes structural defects in the outer layer of the tooth, that is enamel. Now, coming to the classical features of amelogenesis imperfecta, there are two classical features. The first is that it exhibits complex inheritance pattern. Now, what do you understand by the term complex inheritance pattern? You might have heard of many genetic disorder which possess single inheritance pattern. Either they are autosomal dominant type, autosomal recessive type, X-linked dominant type, X-linked recessive type, or Y-linked dominant type. Amelogenesis imperfecta is that genetic disorder which can exhibit either of these forms, either in autosomal dominant type, autosomal recessive type, X-linked dominant type, and X-linked recessive type. It can exhibit in either of these forms. Okay, now coming to the next classical features of amelogenesis imperfecta is that it is independent of systemic conditions. As I have already mentioned in the previous slide, that the enamel hypoplasia is either genetic in origin or environmental in origin. If the enamel plasia is environmental in origin, only then it is associated with systemic condition. Since amelogenesis imperfecta is purely a genetic disorder, it has got no influence of the systemic conditions on it. So now let's look into the cause of amelogenesis imperfecta. Amelogenesis imperfecta is caused by mutations in the genes which encode for enamel proteins. Mutations, since it's a genetic disorder, mutations in the genes causes amelogenesis imperfecta. Now here is a small diagram of a chromosome. This is a centromere, these are chromatids, and this is small arms of chromatids are known as P arms, long arms are known as the Q arms. Now we all know that chromatids are formed by DNAs. Okay? Now this DNA is made up of segments known as genes. So now these genes are responsible for enamel protein formation, proteins which are necessary for enamel formation. Now these proteins, they help in enamel formation. This is a brief explanation of how genes play a role in enamel formation. Any defect or any mutations in the genes can cause abnormal enamel protein formation, which automatically results in abnormal enamel formation or certain defects in enamel. So, this is a brief explanation of how genes affect enamel formation. Okay, now before I discuss the different types of genes which are involved in enamel formation and mutations within which can cause amelogenesis imperfecta, let me first brush up the topic amelogenesis. In order to understand amelogenesis imperfecta, you need to have the clear picture of amelogenesis or process of enamel formation. So here I have a small diagrammatic illustration showing the process of enamel formation. Amelogenesis is divided into three stages. The first stage is the pre-secretory stage, the second stage is the secretory stage, and the third stage is the maturation stage. Let's discuss all these stages in detail. In the first stage, that is the pre-secretory stage or the inductive stage, the inner enamel epithelial cells they transform into ameloblast. Now, ameloblasts are the enamel forming cells. Okay, so only this takes place in the first stage of amelogenesis. This stage is followed by the secretory stage. Secretory stage is the stage wherein enamel matrix is laid down. Now, enamel matrix is constituted or is formed by organic matter or enamel protein. It is in this stage that full thickness of the enamel is established. Secretory stage is the stage which determines the thickness of the enamel. Okay, any defect or abnormality which affects this stage can result in improper thickness of enamel or defects in the thickness of the enamel. Okay, now this secretory stage is followed by the maturation stage. Now, maturation stage is divided into two stages. The first stage is the transition stage, and the second stage is later maturation stage. In the transition stage, few number of ameloblasts undergo programmed cell death or apoptosis, and in the later maturation stage, the ameloblast, each cell of an ameloblast undergo a process known as modulation. Modulation is a cyclical transformation in the distal portion of ameloblast from ruffle ended like this to smooth ended, and then from smooth ended to ruffle ended in order to secrete lysozymes, proteinases. Now, these enzymes help in degradation of organic matter. Degradation of organic matter, which was laid down in the secretory phase of amelogenesis, is very important for proper mineralization. If there is certain defect in this stage and degradation of organic matter or enamel proteins does not take place, then improper mineralization is achieved or improper calcification is seen. Now, this is a brief overview of all the things happening in amelogenesis. You need not remember everything, but you need to remember three main important points, and that's in the first stage, that is in the pre-secretory stage, the cells of inner enamel epithelium they transform into ameloblasts. Like any defects in this phase can result in decrease in the number of ameloblasts, okay, which will later on can cause enamel defects. Now, coming to the next point or the second point is the secretory phase. This is the phase wherein enamel matrix is laid down. The ribosomes on the endoplasmic reticulum they secrete the proteins which are later stored in Golgi complex. These are secreted through the Tomes process, and the organic matter is laid down or the enamel matrix is laid down. It is in this stage where the full thickness of the enamel, where the thickness of the enamel is determined. Now, this secretory phase is followed by the maturation stage. Maturation stage is the stage wherein degradation of organic material takes place in order for mineralization to happen. So, now let's move forward to different types of genes which are involved in amelogenesis and mutations within which can cause amelogenesis imperfecta. Now, the first gene, MLX, is present on X chromosome and it encodes for the protein amelogenin. The next gene is ENAM. It encodes for the proteins enamelin. These two genes are very important for enamel matrix formation. Okay, they help in formation of enamel matrix. So these two genes encode extracellular matrix protein, which is enamelin and amelogenin of the developing tooth, in order to lay down enamel matrix. Now next two genes are kallikrein 4 and matrix metalloproteinase 20. These two genes encode for enzymes which are responsible for degradation of organic matter. If any mutations in these genes result in improper degradation of organic matter, which eventually causes improper mineralization. Okay, now coming to the fifth gene, that is SLC24A4. It is a solute carrier encode for proteins which act as calcium transporters, which help in transportation of calcium during enamel maturation or during enamel calcification. Any defects in this gene or mutation in these genes can cause hypocalcification of enamel. Okay, you need not remember all these genes in detail. All you need to remember is the first two genes, that is MLX and ENAM. These two genes, the mutations in these genes are the most common cause of amelogenesis imperfecta. Now let's move forward to the different types of amelogenesis imperfecta. Amelogenesis imperfecta has been classified based on the stage of enamel formation which was affected, as well as based on the clinical features, how the amelogenesis imperfecta appears. Now first type of amelogenesis imperfecta is the hypoplastic type of amelogenesis imperfecta. It is, now, with the name we know, hypoplastic is coming from the word hypoplasia, which means decreased number of cells. Okay, so this type of amelogenesis imperfecta is caused by decreased number of ameloblast. Decrease in the number of ameloblasts would result in decrease in enamel matrix. Okay. Now, let's look into the features of a hypoplastic amelogenesis imperfecta. Enamel is abnormally thin, of course, because since there is decrease in number of ameloblasts, there is decrease in the enamel matrix. As we have already studied that when the in the second stage of amelogenesis, in the secretory phase, when the ameloblasts lay down the enamel matrix, it is that stage which defines the thickness of the enamel. And if there's some defects, then if there are certain defects in enamel matrix, then the thickness of the enamel is altered. So over here we can see that since there is decrease in the number of ameloblasts and decrease in enamel matrix, the resultant enamel is thin. Okay. Now coming to the next features of hypoplastic amelogenesis imperfecta is that it is hard and differs from dentin radiographically as normal. Of course, since the first two stages of amelogenesis have been affected, which has got no influence on the hardness of the enamel, until then mineralization didn't take place. Mineralization starts only in the third phase of amelogenesis. Since only the first two stages are affected, the hardness of the enamel is not altered, and you can clearly differentiate between enamel and dentin radiographically. Now, this hypoplastic type of amelogenesis imperfecta can exhibit as rough surfaces, pitted surfaces, or smooth surfaces. Okay, but there is subsequent changes in the color from yellow to brown, yellowish brown color, because enamel is abnormally thin and dentin can be seen through. Hence, there is changes in the color of the enamel. Now coming to the next type of amelogenesis imperfecta, that is hypomaturation type of amelogenesis imperfecta. With the name we know decreased maturation, which means that during the third stage of amelogenesis there is certain changes which causes hypomaturation of ame- hypomaturation type of amelogenesis imperfecta. Now let's look into the features. Enamel is of sound thickness. Of course, the thickness was defined in the second stage or the secretory stage of the amelogenesis, whereas hypomaturation or maturation takes place in the third stage. So the first two stages of amelogenesis are not affected in this type of amelogenesis imperfecta. Only the third stage is affected. Hence the enamel is of sound thickness, less hard, of course, because mineralization is improper mineralization, prone to rapid wear, radiographically not as radiopaque as normal enamel. Let's see clinically how does it appear. The hypomaturation type of amelogenesis imperfecta gives a classical presentation of chalky white lesions. This is because of improper degradation of organic matter or enamel protein, which causes defective mineralization or improper or poor mineralization, giving such giving rise to such chalky lesions. Now coming to the third type of amelogenesis imperfecta, that is hypocalcification type of amelogenesis imperfecta. With the name, we know that it is again affecting the third stage of amelogenesis, that is the stage of maturation, wherein calcification takes place. Okay, now let's look into the features. Enamel is of sound thickness because the third stage is getting affected and not the second stage of amelogenesis. It is brittle because of lack of calcium. Is prone to rapid wear, okay, exposing the dentin. Radiographically, same approximate radio density as that of dentin, which is not normal. Since there is poor calcification, you cannot differentiate between enamel and dentin radiographically. And as you can see, since there is improper calcification, the resultant enamel it's so defective that it appears as a dentin. Okay, now let's relate different types of amelogenesis imperfecta with different stages of amelogenesis. The first type of amelogenesis imperfecta was hypoplastic type of amelogenesis imperfecta. Okay, now this hypoplastic type of amelogenesis imperfecta can occur either due to alteration in the pre-secretory stage, that is when there is transformation of inner enamel epithelial cells into ameloblasts. If there is certain defect which can lead to decrease in the number of ameloblasts, automatically there is decrease in enamel matrix formation, which results in thick thin enamel, which is a characteristic feature of hypoplastic type of enamel hypoplasia. There's one more cause for plastic type of amelogenesis imperfecta, and that's in the second stage, that is in the second stage of amelogenesis, the secretory stage, wherein the enamel matrix is laid down. Now we all know, as we have studied recently, that the two genes which encode for enamel proteins are enam and MLX. Mutations in these genes can result in abnormal formation of enamel proteins, okay, which again causes hypoplastic type of amelogenesis imperfecta. Now coming to the second type of amelogenesis imperfecta, and that's hypomaturation. Hypomaturation type of amelogenesis imperfecta results when there is improper degradation of organic matter, which results in improper mineralization. If the degradation of organic matter does not take place properly, then hypomaturation types of amelogenesis imperfecta results. Now, as we have already studied, that the two genes kallikrein and matrix metalloproteinase these are the two genes which are responsible for degradation of the organic matter. Mutations in these genes can cause abnormal degradation of the organic matter, which results in hypomaturation type of amelogenesis imperfecta. Now, coming to the third type of amelogenesis imperfecta, that is hypocalcification type of amelogenesis imperfecta, when there is faulty calcium transportation or block in calcium transportation to the mineralizing enamel. As we have already discussed, that SLC24A4, this is the gene. It encodes for calcium transporter proteins which help in transferring calcium during the mineralizing stage of enamel formation. Alterations in these genes or mutations in these genes can result in loss or lack of calcium transport to the mineralizing enamel, which results in hypocalcification type of amelogenesis imperfecta. Now coming to the fourth type of amelogenesis imperfecta, that is hypomaturation hypoplastic type of amelogenesis imperfecta associated with taurodontism. It exhibits combination features of type 1 and type 2 with taurodontism. Taurodontism is evident in this type of amelogenesis imperfecta. Now, coming to the clinical features of amelogenesis imperfecta. You can see abnormal color yellow, brown, or gray because it's obvious that the enamel is defective. Its grayish white appearance of the teeth is because of enamel. And if the enamel is faulty, of course, the color of the teeth would be altered from yellow, brown. As you can see over here, the color is altered. Okay, it could be pitted or smooth or rough defects. It exhibits in these three types of defects. As you can see, it's pitted over here, pitted and rough over here, and it's smooth over here. Okay, gives chalky white appearance, which is a classical feature of type 2 amelogenesis imperfecta. Chalky white appearance determines the hypomineralization. Okay, increased risk of dental caries. Of course, enamel is resistant to dental caries, and if there's defects in enamel, then teeth are more prone to dental caries. Hypersensitivity, as the teeth are, as the enamel is not as hard as normal enamel, it tends to wear off. Hence, the teeth are exposing dentin. Hence, the teeth are hyposensitive. Rapid attrition, because the teeth are very brittle. Lack of calcification and lack of mineralization makes them really brittle. Hence, they undergo rapid attrition. Excessive calculus deposition, and at times, amelogenesis imperfecta is associated with anterior open bite. So, clinically, most commonly you would see pitted appearance or rough surfaces with pitted appearance. Enamel is thin. There are changes in color, or you can see such bands of hypocalcified or hypomineralized areas, chalky white, giving chalky white appearance. Now, coming to the histological features of amelogenesis imperfecta based on different type of amelogenesis imperfecta. In the first type of amelogenesis imperfecta, that is the hypoplastic type of amelogenesis imperfecta, you see defects in matrix formation. The defects could extend to a point that there is complete absence of matrix. This is a type 1 amelogenesis imperfecta, how it appears under microscope. Coming to type 2 amelogenesis imperfecta, you see defects of matrix structure and of mineral deposition. Okay, this is wherein the abnormal enamel matrix is laid down. Okay, you would see defects in the matrix structure or enamel matrix. In the third type of amelogenesis imperfecta, that is the hypomaturation type of amelogenesis imperfecta, you see alterations in the enamel rod or rod sheet structures because of improper mineralization and calcification. Okay, now coming to the treatment part, there is no obvious treatment for amelogenesis imperfecta. It can only be improved by cosmetic appearance. I hope you all enjoyed this lecture. I hope it should be beneficial for you all. In my next lecture, I'll be talking about dentinogenesis imperfecta. If you like this video, please like, share, subscribe, and yeah, don't forget to hit the bell icon to be updated with my latest videos. Thank you, everyone, and have a nice day.